or breaking apart the chain, taking away, removing one end of it or disassembling one end of it.
Microtubules provide tracks along which membrane-bound vesicles travel to and from the plasma membrane.
That's the motor protein.
The directional movement of these cargo vesicles is due to a family of motor proteins linking vesicles to microtubules. Membrane-bound organelles like mitochondria are loosely trapped by the cytoskeleton. Mitochondria change shape continuously, and their orientation is partly dictated by their interaction with microtubules. All the microtubules originate from the centrosome, a discrete fiber.
All right, I just wanted you to get some sense
of vesicles and microtubules.
It's quite remarkable.
Membrane-bound organelles like.
So, in the CNS, it is the case that, in general, about 10% of the cells that make up what is called the neuropile. Let me write that word out since I don't know if it crops up anywhere else. And actually, I'm not sure that I should be a little more precise in my definition. The neuropile consists of everything in the CNS except cells. So that would include fibers and supportive cells and blood vessels and so on. All right. So the types of cells that are present in the CNS include neurons, and the neurons comprise about 10% of the total number of cells in the CNS, again, depending upon the region of the brain, region of the CNS. The remaining 90% of that tissue consists of glial cells, sometimes called neuroglia. And so we're going to turn our attention to the glial cells now. I've listed at the top here the major categories of glial cells. You can see some are native to the CNS and at least one to the PNS. And they serve a variety of functions. In the illustration here, for example, you can see that the yellow things are axons. We're in the CNS. This picture applies only to the brain and cord. And so these are axons, and you can see that the myelinating cells of the CNS are not Schwann cells. They're cells called oligodendrocytes or ligodendrocytes. Anybody remember from their high school? What did you remember? When they were talking about government, did you ever? Yeah, what was another document? Remember?
Two.
Rule by the few, not necessarily two. Rule of the few. So the idea here is dendro just means branches. These cells have the ability to myelinate a few branches. Oligodendrocyte. They're also called, it helps, they're called paddle cells. I bet you can figure out why. They have a paddle-shaped cell body. But the key thing to remember about oligodendrocytes is they are individual myelinating cells of the CNS only that can myelinate multiple axons. Remember, a Schwann cell can't do that. When we talk about you're going to be studying MS and other diseases, demyelinating diseases, this is the cell that is not functioning properly in those conditions. In this illustration, we also see another type of CNS glial cell called the astrocyte. The astrocyte gets its name from its star-shaped appearance. Looks a little bit like a multipolar neuron, but they're much smaller, has multiple processes. And they serve a variety of functions. We're still learning about astrocytes, or you can find books written about astrocytes to suggest that they're really the most important cell of the CNS. The neurons are comparatively unimportant, according to those theorists. But in any case, the astrocyte in this illustration is contributing to what is called the BBB. What is the BBB? The blood-brain barrier. Now we're learning that the blood-brain barrier isn't so much of a barrier as we once thought. But if you look at the relationship or the position of this astrocyte in the CNS neuropile relative to the axon, and this thing, of course, is a cerebral capillary, you'll notice that it's interposed between the two, which means that, and as a first approximation, it's taking nutrients from the cerebral blood, from the cerebral capillaries, processing it, if you will, and then making it available to the axonal process. So it's acting as an intermediary to prevent the vulgar systemic blood from tainting the pristine neurons. It's acting as, again, part of this blood-brain barrier that we will address in a little more detail later. Yeah. Well, there's going to be, as I say, we don't see it here, but as part of that neuropile, whatever color this is, there's going to be a profusion of other blood vessels. And you can imagine that not all blood vessels in the brain are going to be involved with astrocytes. So these cells are going to have capillary vessels near them, and they'll be able to extract the nutrients in a manner we'll talk about when we talk about the cardiovascular system in much the same fashion. Yeah, these cells aren't so picky, but at least in theory, the neurons are quite picky with respect to what type of nutrients and other substances they can acquire from the systemic blood. So I've listed here some of the major functions of glial cells. We've already talked about myelination. This is the role of the oligodendrocyte in the CNS, the Schwann cell of the PNS. The specialized immunological cell of the CNS is something called the microglial cell, microglial. So I guess the reason I begin a discussion of this slide in that fashion is most of these functions then apply to astrocytes. Astrocytes seem to be the principal figure. They contribute to the blood-brain barrier. They contribute to organization, developmental organization of the CNS. Remember, you started out as a zygote, a single cell, and then you became basically 3 tissue layers. And then from that, you just think about the CNS alone. If you get a chance, we won't have time too much in here. Do a search for connectome. We had it in the previous, we talked about it this morning. And look at the remarkable organization of white matter pathways in your brain. They're surprisingly linear. It's not as if the white matter pathways in your brain, one's going this way, one's going this way, but they're very organized. How does all that happen? How do the axons know exactly where to go? Well, we know that there are a lot of trophic factors involved developmentally, but also these glial cells seem to play a very important role. When the glial cells are not expressed properly, a lot of that differentiation and specialization of tissues in the CNS tends to go awry. The astrocytes also help to facilitate the CNS response to trauma and injury. And in some cases, that's very beneficial. For example, microglia has the ability to deal with infective agents and possible autoimmune problems. But the astrocytes take a different approach. They say isolate the aggressor, isolate the intruder, wall them off, and prevent them from doing any further damage. And sometimes that can have adverse consequences. In fact, the astrocytes contribute to, I don't know if I have it up here somewhere, scarring. They contribute to CNS scarring after some types of trauma. Yeah, there we go. Remember when I asked you to count all the ways that neurotransmitters at a synapse were being removed from the synapse. We saw that in some cases, the neurotransmitters would be taken into peripheral capillaries, but in other cases, the neuroglia help to regulate neurotransmitter levels. So again, especially the astrocytes play a very important role in regulating the concentration of neurotransmitter in a region. It says down here there are many more glial cells. I said about 10 and 90%. Varies from region to region. There are certain regions of the brain where the ratio is about 1 to 1. Other regions, it's about 9 or 10 to 1, especially in portions of the brainstem that dramatically outnumber the neurons. So you have several slides here where you can see the astrocytes. The astrocytes are the little cells, they're star-shaped cells. We've talked about the function, their ability to regulate neurotransmitter release and uptake. We talked about glial scarring. In this graphic, these would be the neuronal processes here. These would, the little black things would be the astrocytes. Here you can see a neuronal cell body up in here. The microglia, the specialized immune system, the immune cell of the CNS, also has its heckle and jive. Heckle and jive. Heckle and jive. Somebody help me out here. I'm having a senior moment and you're just laughing. What is it? Thank you. Somebody just ought to put me out of my misery. All right. They too have, I'll make it simple, Janus face to nature. Remember Janus face, the two sides of the Greek, whatever it was? Good side and bad side. So these microglia are thought to be specialized monocytes that migrate from the general circulation, take up a home in the CNS, and then take on this morphology here. They act to, again, defend the CNS against a variety of potential intruders, everything from bacteria to autoimmune problems. But they too have a sort of hyper-reactive response. You can see over here, these are activated microglia. They tend to aggregate wherever there's a problem. This is part of the inflammatory response, which, as you know, is not always helpful in the long run. For example, you're going to learn about so many different types of dementia later, everything from Tick's disease to Lewy bodies to the amyloid plaques and Alzheimer's disease. And where those protein accretions develop in the brain, and by the way, studies show they're present even in your young brains already. This is just part of being human. These things tend to aggregate around them, and once again, they tend to try to destroy them. And that's fine. You think that would be a good thing, except in the process, they often take other viable nervous system cells with them, neurons with them, and glial cells with them. So activation of microglia usually comes at a cost, and there are even therapies that you will learn about later intended to minimize, to reduce the activation, the inflammatory response of the microglia because of the potential adverse consequences.
I think I blacked out. Did you say microglia are also astrocytes?
If I did, I'm sorry. They are glial cells. I remember something about Jekyll and Hyde. But, everything went black over that. Now, I put this little link up on the website about this transformative discovery. I was talking this morning about the fact that the immune system is heretofore thought to be immunologically privileged. But this may change everything. Again, this one, what's the 2015, received October, published in June 2015. And they argue, I think I have an example here. This is the structure you learned today, the superior sagittal sinus. So what we're looking at in those red regions there are the putative lymphatic vessels that run along the superior sagittal sinus. If I had suggested such a thing prior to this discovery, it just would have been laughed off the stage, laughed off the classroom. And of course, there's still some debate about this because this is not in a human brain, this is in a rodent brain. Some of us do fine with rodent brains. I've been working like this way for years, but it's not evident that these structures are present. The authors of the study contend that they are. And the graduate student came up with this very, very interesting process where he just found that these structures, they weren't necessarily cellular structures, were just flowing along certain pathways, and that he used a variety of stains until eventually those pathways became visible, and it turns out they appear to be lymphatic vessels. In any case, this could have implications for what I was just talking about. A lot of the diseases of the nervous system involve accumulation of proteinaceous plaques. Like plaques, in this case, we use the word variety of ways. But in this case, we mean accretions, just globs of protein that don't belong there. This is true for Alzheimer's. This is true for, did you read this week about the potential new therapy for Creutzfeldt-Jakob and squamous prion diseases, right? What are prions? Yeah, prion diseases are when, for whatever reason in your brain, it's usually in the brain, a normal protein changes its conformation or shape. It goes from a pleated sheet structure to some abnormal structure. And when it does that, for whatever reason, other proteins around it begin doing the same thing. They're no longer functional in whatever tissue they are, and then the immune system starts attacking them. And these are some of the things we've talked about. So, just to sort of wrap two different things up here. There are tribes. I don't know what they are. It's the Foray tribe in Indonesia, is it?
Also New Guinea.
Thank you. That have, as part of their cultural practice, they eat the brains of their loved ones. I guess that's a nice way to say that. And initially, they would get a disease called Kuru, which is another one of these prion diseases. But some of them became resistant to Kuru. And so now, what is the potential therapy for all of these prion diseases? You're the investigator, you're the physician. You've now found, and by the way, a famous person just died of this. Head of a tech company just passed away from they call Creutzfeldt. But anyway, based on what we just said, how would you work on therapy for this problem? What information do you now have that might help you?
Try to find like what proteins they express that help like fix the prions.
Okay, in some cases we already know that. We know a lot about the prions, but we had no way to. These diseases are in very good field. There's no cure. But now, anybody have any ideas? Yeah, there you go. Let's see if. It's a kind of grisly way to get an antibody, but yeah. So in any case, getting back to where all this started, if it's true that this system is present in the brain where we didn't think it was, and of course, what does the lymphatic system do as part of the immune system? What does it tend to do? What's its general function? It has a lot of functions. Of course, its job is to address the accumulation of tissue, of structures, tissues, and other substances in the body that don't belong there. Well, amyloid, Alzheimer's disease, and such a thing. Maybe you could now target the system to upregulate it in some fashion. In any case, we now know it's there, if indeed it is, and that could possibly provide another avenue for treatment of a variety of dimensions. So we talked about oligodendrocytes. Again, the key things to remember here, these are the myelinating cells of the CNS. They have the ability to myelinate up to 50 additional. I'm sorry, Luis, did you have a question?
It's not.
Good. That's what I like to hear. And in the brain, remember, these are the cells that contribute to saltatory conduction. We talked about saltatory conduction, and you can see they do it a little differently. They have these regions of myelination, of insulation. The nodes of rendezvous would still be between those regions of myelination. The Schwann cells, conversely, are the myelinating cells of the peripheral nervous system, and they work by an entirely different principle. You can see they sort of begin as a single sort of a cuboidal cell, and then start wrapping their cytoplasm and neurolemma. Remember, the neurolemma is the cell boundary around the axon, until eventually all that's left is this laminar organization of neurolemma and a peripheral Schwann cell body. We also talked about these things. Remember the Remac bundles. Technically, we describe a Remac bundle as a non-myelinating Schwann cell. what seems, its primary function seems to be simply to disaggregate or separate the axonal processes from each other. We talked about these cells this morning as well. Now, these are the ependymal cells. And these are ependymal cells, these dark staining cells that you see here. This is in a healthy central canal of the spinal cord. Remember, the central canal is in that, where the two wings of the gray matter, the two butterfly wings, right there where they come together in the middle of the spinal cord. Well, the CSF is flowing through that central canal. And these ependymal cells seem to be pretty much the default cell of the ventricular mucosa and the central canal mucosa. And they're very, very specialized, remarkable cells. They sort of violate one of our basic principles of cell biology. They have both cilia and microvilli. They have both cilia and microvilli, which means they are doing that. They're pumping the CSF along, sort of facilitating that pressure gradient where the pressure gradient is going to be very low. But remember, their other function is to regulate CSF volume. And that's what the microvilli are doing. The microvilli are both secretory and absorptive. But in ways that are only partially understood, the ependymal cells contribute to the proper volume of CSF. We talked about axonal reactions. I don't think I need to talk too much more about that. You know about Wallerian degeneration. You know about the propensity or the proclivity of the proximal neuron once the axon has been severed to form what is called a growth cone in the absence of the myelin sheath. And we talked about the fact that depending upon, you can see macrophages here coming up and cleaning up the mess, cleaning up the debris after the injury. We talked about the fact that depending upon the function of the neuron, the distal neuron, if it can't find another line of work, sometimes it can, depending on the location. Let's say it's a motor neuron, it's going to degenerate and undergo Wallerian degeneration. The associated muscle cell may also do the same thing. And then again, depending upon a variety of factors, even the proximal neuron over a greater period of time may also degenerate. I've made reference to the term trophic factors on a few occasions. Trophic factors are basic. The word trophic just implies nutrition. It's used in two different ways, but I guess they're related. Trophic is used to imply eating or nutrition, but it's also used in place of the word tropic, which is actually a different term, in that tropic means movement toward, growth or development towards something. So when we talk about trophic factors in the CNS, we're generally talking about a variety of chemicals, a variety of substances that serve a number of functions. In mechanical trauma, such as you see here, trophic factors seem to be very important in the ability of this neurite to regrow. And it seems that if we can put the trophic factors in the proper location after this type of injury, that dramatically increases the likelihood of recovery.
How are olfactory nerve cells affected? Can those regenerate?
Yeah, olfactory nerve cells are remarkable in a variety of ways, and there are several types of olfactory nerve cells. But certainly, what we call the primary neurons, we've already defined what a primary neuron is. Yeah, those have some limited ability to degenerate or to regenerate. Problem is, and I think we talked about this before, they're among the first cells to go in any type of nervous system disease. So you have a curious antinomy there. Remember, you said that loss of smell is often an indicator of dementia, and often one of the earliest indicators. But in any case, trophic factors are important in sort of the recovery process, and that's sort of consistent with their ability both developmentally and in recovery from trauma telling the neurites, telling the axons, telling the dendrites exactly where they need to go. And so trophic factors developmentally are absolutely critical for differentiation and development of the normal brain and CNS. I guess that's about all. There are any number of different trophic factors. You can see some of their NGFs and BDGFs, their growth factor and brain-derived growth factor. But all of these are very, very important in the ability of neurites to proceed along the desired anatomical pathways. And we talked about this a little bit a few days ago as well. Remember I said that in the spinal cords of most vertebrates and most higher vertebrates, there are intact, complete motor pathways and programs, such as again, if you remove the cortex from a cat or a rat, people still have the ability to walk or ambulate. You've probably heard the phrase, if indeed you haven't observed a chicken running with its head cut off. You say, okay. I had to do that when I was a child. I'm being emotionally savage to this day. But that's because that motor pathway, that ambulatory motor routine, is present in the spinal cord. The brain doesn't need to be involved with these basic pathways. Now, humans are a little bit different. We seem to have less of that. But what's happened in a case like this, and to make sure you understand what we're saying here, this was a spinal rodent. By that I mean the spinal cord was severed. And they didn't reattach the spinal cord. They didn't reattach the proximal and the distal halves after severing. What they did is tapped into the distal cord and gave it electrical signals, gave it timing signals. And this seems to work in humans as well to some extent, because there was enough of that motor pathway, that motor routine in the cord, that if you could just restore the timing signals, that might be sufficient. Electrical stimulation. Of course, that's a much more sophisticated process in humans than it would be in a rat. By the way, neuroplasticity, anybody want to define that term? What do we mean by plasticity of the brain? CNS.
Shape?
Yeah, in a not so much in an anatomical sense, although that would be important, as in a functional sense. Generally, the idea is that if you lose, for whatever reason, let's say following a stroke or a tumor, whatever, you lose brain tissue, and you lose the ability to perform certain tasks associated with that brain tissue, then often it is the case that surrounding brain tissue will have the ability to recover those tasks. They just simply rededicate themselves, the tissue rededicates itself to the functions that were lost. So plasticity is remarkable. You're going to have stroke patients who, for a time, can't move a portion of their body, and it slowly comes back. They have a stroke, and maybe they've lost a portion of their sensorium. They gradually recover. It may or may not occur. There's no guarantee that will occur, but that's the notion of plasticity. I just thought this was curious. Anything odd about this? As a PA, this would attract your attention? Anything out of place here? And so I'm asking you what brought this fellow to the hospital. He drove his plow truck for 8 hours. After this happened, took his kids to a play rehearsal, took a nap before he went to the doctor. Of course, we'd have to replicate this to prove that it actually happened. If anybody had anything.
So what was the chief complaint?
Well, I mean, clearly he wasn't in a great deal of pain. Of course, you should be able to provide an explanation for that, right? There's not going to be a lot of pain or something. I think if you find the article, this has happened several times. People, for some reason, have a remarkable ability to nail their heads with nail guns. But you'll find all kinds of studies. But what would have brought him to the hospital? He was having some mild headaches.
Yeah.
And he didn't even remember doing it. He remembered itching. He heard the gun go off. He felt a little blow. He thought the nail went beside his head or something, and the gun was just. He had no idea that he had a nail in his brain. Of course, what's the real problem now? You're the PA.
Taking it
Getting
out.
it out. You're going to do more as much damage getting it out as just leave it in there, I guess. Don't worry about that one. That was what I was working on. By the way, though, it says here, this is a new study regarding sleep. Again, need to be responsible for this. When do you've been studying, when do you learn? At what point in the day do you do most of your learning? Yeah, while you're asleep. And so again, that redounds the importance of sleep. Sleep is also important for vigilance, the ability to monitor the world around you and be able to understand what's going on around you. But this is a recent study, and notice what they said. They showed that again, alt AD is more likely to occur. This is an elderly population, but it's more likely to occur, especially the symptoms of Alzheimer's, in patients with impaired sleep. The study has revealed that people who get regular sound sleep for at least 8 hours a day are at decreased risk of suffering from AD and losing their memory. So there, sleep is very, very important. All right, so a lot of this material, we're going to talk a little bit about the anatomy and the organization of the CNS. Unfortunately, we've already talked about a lot of this in the anatomy course. I'm going to zip over a lot of this, but just make sure everybody's on the same page. It was clear to me from the test results today that most of you are extremely capable. I'm going to have to work very hard to come up with a question that will gratify me and not you. I'm joking, of course. I'm happy when you're happy. But some of you need to help each other, and I'll be happy to help. You need to make sure everybody understands these general principles we've been talking about, because they're central to the remainder of your training. So make sure you know the organization of the spinal cord here. We talked about the butterfly organization of the gray matter in the cord. We talked about the white matter peripheral to that. The dorsal horns are sensory. The ventral horns are motor. You should be able to describe for me the structural categorization of these neurons that you see in the circles here, the red circles that I put up there. You should be able to tell me what the words efferent mean, so on. We didn't talk too much about this in the anatomy, but remember, the spinal cord is considerably shorter than the spinal column. The spinal column goes from your head all the way down to your butt, down to the coccyx. But the spinal cord ends where? What is your spinal cord in? Yeah, just down about L2 or so. It depends on how tall you are and various other things. So that's amazing. But your spinal cord stops at about L2 and is usually less than 20 inches long. And that's going to depend upon how tall you are and so on. And so we need to talk about the anatomical consequences of that. I wonder if I have an illustration here, just again in my effort to abbreviate the slides to accommodate the brevity of this course. We just don't have time to talk about much anything in detail. You should be familiar with the cervical and lumbar enlargements of the cord. The cervical and lumbar enlargements of the cord. And by that we mean that the diameter of the cord is greater in the cervical and lumbar regions. Why would you suspect that is the case? Because at the, yeah, did you have that's fine, no, please. Exactly right, you're exactly right. There's going to be an efflorescence of fibers emerging from the cord at these levels, one for the upper limb, one for the lower limb. Now, in general, the cord is tapering off and getting smaller, so this is, when we talk about these enlargements, these are relative to the superior diameter of the cord. Just a little more terminology here. We talked about most of these concepts. If you look at this section of the cord here, you know that the dorsal side is at the top, right? And the ventral side is at the bottom. We know that in general, we can sort of draw a line there and sort of imagine that at least the dorsal gray matter is going to be sensory. The ventral gray matter is going to be motor. Oops. And while that's true, unfortunately, that's about as much of a generalization as we can make. Now, in between the dorsal horns are these things, which we talked about previously, being the dorsal columns. And later, when we get to the sensory unit, you're going to have to learn the dorsal column medial lemniscus sensory pathway.
So the CSF will be running through.
And then around it as well, in the subarachnoid space. So those are the dorsal horns. But what I want to direct your attention to now is the white matter. So this would be all of this white matter out here would be sensory. These are afferent pathways. By afferent, the signal is going that way, right, going toward the brain. But as I said, that's when our ability to topologically or geographically assign function tends to break down. Because indeed, all of this blue that you see here in the white matter, those are all afferent pathways. Those are all sensory pathways. And indeed, look where the major motor pathways are located, right up in here. So I want you to know this one. We talked about the dorsal columns, which are sensory. You should know that. Dorsal columns are sensory in between the dorsal horns. This track here and this track here, this forms the corticospinal tract, or at least the largest part of the corticospinal tract. Now, pay attention to the prefix and the suffix here, corticospinal. The general rule is, if the suffix is spinal, corticospinal, we're going from the brain to the spinal cord. Is that motor or sensory?
Motor.
Right, so we're going to, whereas if I'm talking about the, let's say the spinothalamic tract, would that be sensory or motor? Sensory, because the spinal is the first. So you get the idea. Based upon that little rule, you should be able to designate any pathway, any of these tracts as either motor or sensory. So if the ends in spinal, it's going to be a motor tract. And this is the most important one, the corticospinal tract. Also called the pyramidal tracts. The corticospinal tracts are the pyramidal tracts. And we'll see why that is a little later, but it has to do with how they appear, how they manifest in the brainstem. Any motor tract that is not part of the corticospinal system is called extrapyramidal. In this case, extra just means outside of. So an extrapyramidal tract you should be able to deduce is a motor pathway that is not part of the corticospinal pathway. And there's several of these. There's a very old postural pathway that's not so important in humans. We think that the primary function of the rubrospinal, rubro means referring to the red nucleus, is to help you maintain posture. Right? You're not thinking about your posture too much, but that helps contribute to that, at least in primates. These tracts are more associated with the cerebellum and contribute to, we'll talk a little bit about the cerebellum later. But for now, I just want you to, as you look at this transection, to pay attention to the relative position, the relative organization of the sensory pathways and the motor pathways. We'll talk about these two major sensory pathways later, and you're going to have to learn the organization of these pathways. So I'm not going to ask you to worry about that now, except you should pay attention again to the two names that are most important. This is the dorsal column. This is the dorsal column pathway up here in between the two dorsal horns. The other one that you're going to have to pay attention to later is this anterolateral system, which is sort of diffusely organized around here. Both sensory, this one at the very least, in the wrong place, right? We'd all agree it shouldn't be where it is. And then know what we mean by pyramidal tract and extrapyramidal tract. You should know that pyramidal tracts are the corticospinal tracts. These are the tracts that enable you to make very, very precise movements with your fingers and your mouth and so on, to some extent. And then most of these tracts are for more basic fundamental motor functions such as balance, orientation, sort of thing. We've talked about dermatomes over here. Whoops. Over on the right side, you just see a little table of levels at which both motor function and sensory. I think this is related only to motor function, but this is the level at which impairment is lost, depending upon how far down we go to the spinal tract. Notice the very top there. It says if there's injury to the spinal cord, even at C3 or C4, that could involve that could involve your ability to breathe, because that's the motor pathway, the phrenic nerve that goes down to the diaphragm, and you lose that, you lose your fundamental ability to inhale and exhale, more so to inhale than exhale. And then I'm not going to ask you to memorize these things right now. There are a few things I want you to know a little later, but you can just see the relationship with respect, for example, to paraparesis, quadriparesis, and so on as you move down the spinal cord. So why don't we take a break there? We'll come back and talk about some general principles of spinal function.